and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on chromosomal mutations. In our previous discussion, we learned that changes in the arrangement or structure of chromosomes can lead to mutations. We specifically discussed three different types of chromosomal mutations, including aneuploidy, polyploidy, and autopolyploidy. We learned that aneuploidy is a chromosomal mutation that involves an abnormal number of chromosomes. Polyploidy is when cells or organisms contain more than two sets of chromosomes. We also learned that autopolyploidy are polyploids that have multiple chromosome sets that come from a single species. In today's lesson, we will discuss several more chromosomal mutations, including deletion, duplication, inversion, and translocation. All of these mutations involve structural changes or gene rearrangements within a chromosome. Students, if you are prepared, then we will begin. The first chromosomal mutation we are going to discuss is deletion. This mutation causes a decrease in the number of genes due to the deletion of a large section of a chromosome. The chromosome breaks, and part of its genetic material is lost. Chromosomal deletion can result in a wide array of genetic disorders, such as Prader-Willi Syndrome. This condition occurs when there is a malfunction of the hypothalamus. Prader-Willi Syndrome affects many functions of the body, including hunger and satiety, temperature and pain regulation, fluid balance, puberty, and fertility. Other genetic disorders that result from chromosomal deletion include infertility in males, Duchenne muscular dystrophy, and Cri-du-Chat syndrome. Duplication is a chromosomal mutation that occurs when genes are duplicated and are displayed twice on a single chromosome. Often, this type of mutation is harmless because the chromosome still retains all of its genes. However, if the entire chromosome is duplicated, the results can be much more serious. For example, having 3 copies of chromosome 16, which is known as trisomy 16, can result in a range of medical issues. These include poor fetal growth, muscular and skeletal anomalies, congenital heart defects, and underdeveloped lungs. Chromosome 16 is one of the 23 pairs of chromosomes in the human body. This chromosome spans about 90 million base pairs and accounts for almost 3% of DNA in cells. Inversion is a chromosomal mutation that occurs when an area of DNA on a chromosome reverses its orientation on the chromosome. A person can develop leukemia when an inversion occurs on a chromosome 16. Leukemia is a type of cancer that exists in the blood and in the bone marrow. People who have this disease have an abnormal increase in the number of white blood cells in their bodies. Since the cells are created so quickly, a large number of abnormal cells develop and healthy cell growth is impaired. If inversion leads to an embryo having too few or too many copies of genes, then the embryo could be miscarried. It could fail to grow properly, or it could be born with substantial medical problems. The final chromosomal mutation that we are going to discuss today is translocation. This is when a piece of one chromosome is transferred to another non-homologous chromosome. Translocation is often responsible for causing chronic myelogenous leukemia. This is a type of leukemia that is characterized by increased rapid growth of myeloid cells. These cells are found in the bone marrow, and they accumulate in the blood. Students, please list the 4 chromosomal mutations that we have discussed in this lesson and briefly describe each.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, everyone. I am sure that you had no trouble with this activity. During this lesson, we discussed deletion, duplication, inversion, and translocation. Deletion is a decrease in the number of genes that occurs as a result of the loss of a large section of a chromosome. Duplication is a chromosomal mutation that occurs when genes are duplicated and are displayed twice on a single chromosome. Inversion is a chromosomal mutation that occurs when an area of DNA on a chromosome reverses its orientation. Finally, translocation is when a piece of one chromosome is transferred to another non-homologous chromosome. Were you able to describe each chromosomal mutation correctly? If so, you are doing great. If not, please review your notes at the end of the lesson. As we have learned, chromosomal mutations can lead to a number of genetic disorders. For the following activity, please match the chromosomal mutations with the conditions they can cause.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Hello, class. Were you able to match the chromosomal mutations with the conditions they can potentially cause? Deletion can lead to Prader-Willi Syndrome, Duchenne muscular dystrophy, or Cre-du-Chat syndrome. Duplication can lead to trisomy 16, which is when someone has 3 copies of chromosome 16. This can cause poor fetal growth, muscular and skeletal anomalies, congenital heart defects, and underdeveloped lungs. Inversion can cause leukemia, and translocation can cause someone to develop chronic myelogenous leukemia. In today's lesson, we discussed several different types of chromosomal mutations, including deletion, duplication, inversion, and translocation. We described each of these mutations, and we learned about some of the genetic disorders they can potentially cause. This brings us to the end of our lesson for today. Until next time, thank you, teacher. Thank you, students.